I think you can start and
Okay,
I
okay.
will.
Just a quick presentation about the themes of the workshop, and thank you, first of all, very much for being there. I'm sorry not to be in presence. So, just a few words about the motivation behind this workshop. The title is Molecular Communication Approaches for Wetware Artificial Life, and this is already a second edition. The initiative is between me and Yutetsu Kuruma, which is also another artificial lifer, let's say, and some young people from the world of communication engineering. Uh, because recently I discovered this community which is very much interested in exploiting molecular communication to advance somehow the artificial life or synthetic biology technology. So these are the motivation. As you know, in recent years, there have been several advancements in systems and synthetic biology, but these, of course, promote also the wetware approaches to artificial life. We can use biochemicals or just chemical materials, as also shown yesterday, for example, in the JITCA workshop of ChemAliForms, to build, to construct systems capable of displaying lifelike behaviors. Uh, in addition, thanks to this use of chemical material, these kind of artifacts, they show a deeper phenomenological and organizational relevance when compared, for example, to robots or um, software. In this sense, we believe that wetware artificial life is one of the frontier of artificial life that need to be developed at the maximum extent. And in addition to explore or study the chemical and physical laws that rule the behavior of these systems, we can also focus on information and communication theories. For example, I don't know if you are aware that there is one community in engineering, probably founded by Tadashi Nakano, who independently, totally independently from synthetic biologists, they start to imagine to create a network of objects, let's say, which can be synthetic cells or whatever, that communicate with each other. And there is already, a book, some theories. Actually, they try to translate the information theory, which is typically developed at the logical level, into the material level, in the sense of taking into account stochastic diffusion and all other properties of chemical compounds. In addition, a scenario which for me is quite interesting, even if it's still very premature, maybe, is something called Internet of Bio-Nano Things. Is a net, is a set of things which communicate by chemical messages. So a perspective can be that wetware artificial life can take advantage from this combination, a combination of areas such as synthetic biology, systems chemistry, molecular communication, and so on and so forth, in order to advance conceptual understanding, technological abilities, and applied research. So this workshop series is an attempt to provide an arena for discussing all these topics and try to progress the field. If we speak about synthetic cells, you know that there are several approaches. Let's say the red one, the green, the red, and the blue. In this approach, for example, you just engineer living cells to let them do something useful. For example, to produce fine chemicals, to produce drugs, and so on and so forth. Then there is another approach where you try to minimize the genome of living organisms in order to identify what is the so-called minimal genomes. In this way, you remove what is unnecessary genes, and then what you have at the end is a minimal organism. And third, there is another approach which is completely different, starts from separated molecules. In this drawing, I I report biological molecules, but you can do a synthetic cell, of course, with completely artificial molecules or something that recall the origin of life, for example. And we call this bottom-up approach. Again, in all cases, you create a synthetic cell, but they are quite different in the sense of how they are built and for what, for which kind of questions they are built. And this the idea of communication already generated some interesting perspectives. For example, in 2006, Lee Cronin and several others proposed to investigate what is life by means by a kind of imitation game, like the Turing imitation game. But this time, instead of being done by discriminating a person and a computer, the artificial cell must discriminate between a biological cell and another artificial cell. In 2009, for example, there was a first report of an artificial cell which sent a signal to a biological cell. And there are also some interesting perspectives in applied research. For example, Stefan Leduc tried to put forward the idea of nanofactories. Nanofactories actually are artificial cells which can interact with some biological tissue, and they can sense the environment, they can perceive some input. Inside there are some metabolic modules, and at the end they can produce, for example, a drug. This idea requires a kind of communication between the environment and the artificial cell. So the program for today is a very short program, and I thank again the people which are there. And so first talk will be by Jitka Seikova about innovative methods for selective silencing of microbial signals using microscavengers in artificial life systems. Then there is a talk by Marco Villani and Roberto Serra. Marco Villani will present. And this is about asymmetrical division in population of protocells. Quite interesting because one of the main properties of artificial cells are the capability of dividing. And then there is a talk from Thomas Gorowczowski about programming biological systems with synthetic biology. And then we have a short discussion and maybe some remarks. So before leaving the stage to Jitka, let me advertise also a special issue I'm editing for this journal, IEEE Transaction of Molecular Biological Multiscale Communication. And this is titled Advanced in Synthetic Biology, Artificial Cells and Molecular Communication. You can submit the manuscript by the 15th December of this year. Okay, so thank you very much. Again, I'm sorry not to be there, but